Hi, everyone. I'm Jessica Scott, Research America's Science Policy Fellow. Thank you so much for joining us for our second annual Flash Talks competition preliminary round. The Research America Flash Talks competition is similar in design to the 3-minute thesis, a science communication competition that challenges scientists at universities to present complex research topics and their significance in a concise way. Each of our competitors has prepared a talk describing their scientific research in 3 minutes or less using a single slide, which is no easy feat. Each presentation is a terrific example of science communication, which means our judges, who are tasked with selecting 5 finalists to present at the National Health Research Forum in September, also have a very difficult task today. After all 9 competitors have presented, we will open up the floor to audience questions. But you may type your questions as we go in the Q&A box, just making sure to note which speaker you are addressing. Our judges today are Dana Acton, Director of Science Policy and Legislative Affairs for the American Association for Cancer Research, Dr. Lauren Powell, President and CEO of the Equitist and Vice President for U.S. Health Equity and Community Wellness at Takeda Pharmaceuticals, Dr. Joel Beach, Vice President for Corporate Affairs at ASI Incorporated, and Dr. Kafui Durasa, the K. Rangarama Krishnan Endowed Associate Professor of Psychiatry and Behavioral Sciences at Duke University Medical Center. Thank you, judges, for being here, and everyone, I hope you enjoy the presentations. Our first speaker today is Salvatore Aiello. Salvatore graduated with his bachelor's degree from the University of Michigan with a double major in biomolecular sciences and Spanish. He also has a master's degree in biomedical sciences from Rosalind Franklin University, where he is currently a 5th-year MD PhD candidate. He will talk to us today about his research on resuscitation science methodologies to improve out-of-hospital cardiac arrest survival. And Sal, whenever you're ready, you can go ahead.
Okay, thank you. Cardiac arrest occurs when a person's heart stops beating, and it's fatal if left untreated. While occurring in over 300,000 Americans every year, less than 5% actually survive with good neurological function. So suffice to say, cardiac arrest is a major public health issue. And today I'll focus on one form of cardiac arrest called ventricular fibrillation. And to the left of the screen, you'll see an example of a healthy heart rhythm. And to the right of the screen, you'll see an example of that same heart in a form of cardiac arrest called ventricular fibrillation. And while there are other forms of cardiac arrest, ventricular fibrillation is the only form that requires an electrical shock to restart the heart to a healthy rhythm. And outside the hospital, anyone can deliver a shock using an automated external defibrillator, or you may know it as an AED. And so when someone suffers a cardiac arrest, we are taught to perform CPR by compressing the chest and using an AED to deliver electrical shocks at a set time interval. However, this approach of delivering electrical shocks at a set time interval is analogous to the bygone era of using MapQuest for driving directions. MapQuest directions were static, we printed them out, and they only gave us one route from our starting direction to our end destination. And in the context of treating someone with ventricular fibrillation, our starting point is grabbing that AED and our end destination is shocking them back to life. And of course now GPS systems update in real time and give us turn-by-turn directions so that if the conditions change, our GPS will allow us to adapt. And similarly, our AEDs have voice command technology and the ability to analyze data in real time. But yet we've been unable to customize our CPR approach to an individual. And my research seeks to change that. We use a technique that analyzes the height and frequency of the ventricular fibrillation waveform to measure the amplitude spectral area, or the AMSA. And AMSA is our GPS during CPR because it allows us to assess the quality of chest compressions and precisely time the delivery of an electrical shock to that moment in which the shock has the greatest chance of restarting the heart. So in short, if the conditions change during CPR, AMSA will allow us to adapt. And in our lab, we performed a study that found our CPR approach using AMSA outperforms the standard CPR approach. Animals that had the timing of the shocks guided by AMSA had increased survival and improved heart function. But most strikingly, In the standard approach, there were 2 to 3 times as many electrical shocks delivered to the animals. So what that means is that those shocks were needlessly damaging the heart tissue and not restarting the heart. So what we found is that AMSA can bring CPR out of the antiquated MapQuest era, and we can tailor electrical shocks to an individual. If our findings from the lab can translate to the clinic, we can increase survival by 20%. So I am excited for the role of AMSA in CPR. Thank you.
Salvatore, thank you so much for kicking off the competition with an excellent start. Do our judges have any clarifying questions?
Jessica, it's Joel Beach. Just one very quick one. Salvatore, thank you. That was great. Quick question, has this technique been used in humans yet?
So yes, it is. There's currently a clinical trial going on in Italy. And so that was slightly postponed by the pandemic. So we're eagerly awaiting the results of that clinical trial.
Great, thank you very much.
Mm-hmm.
Thank you, Joel. Our next speaker is Trevor Desmordaunt. Trevor received his bachelor's degree in neuroscience from Brigham Young University in 2019. He is now a master's student in the Department of Neuroscience at Johns Hopkins Bloomberg School of Public Health. Today, he will present his research on biomaterials to improve spinal fusion outcomes. Trevor, you may begin.
Thank you. It's great to be here. More than 65 million Americans report suffering from back pain each year. Most of the time, exercise, physical therapy, and medication are sufficient to manage this pain. However, when medication and therapeutic options fail, spinal fusion surgery is a common solution, with over 1.6 million people every year getting the surgery in the US. This surgery joins the vertebrae around the damaged site together, forming new bone that supports the spine. Spinal fusion surgery relieves pain by joining together or fusing vertebra in a way that removes the pressure on your nerves. Fusion is made possible by the placement of screws and rods that support the spine in the short term, and bone grafts that grow and provide long-term structural support. Bone grafts can come from the patient's own body, known as an autograft, but this is an additional surgery that adds significant time and pain to the patient's recovery. Synthetic grafts of various materials are gaining popularity as a scaffold to facilitate natural bone formation. However, anywhere from 5 to 30% of the time, infection or insufficient bone growth lead to fusion failure and continued pain. Our goal is to make a bone graft that will ensure strong bone formation every time. Here at the Witham Spinal Fusion Laboratory at Johns Hopkins, our work in developing new bone graft materials is already showing promise in this area. We start with a scaffold made of materials that mimic the composition of bone. If we compare the cells in our body that build bone, osteocytes, to a construction crew, our bone graft is akin to a frame and raw materials that those cells can use to build bone. When our cells show up to the construction site in the spine, they can get right to work on putting all these materials together to form strong, new bone. We can further jumpstart construction by adding growth factors to the graft, molecules that direct the cells around the graft to make bone. We can also treat stem cells and implant them with the graft to encourage more robust bone growth. We are currently developing a bone graft loaded with antibiotics to help prevent infection from interfering with bone formation. Through these and other methods, we are on the path to a bone graft that will enable 100% success rate in spinal fusion surgeries. While our lab is focused on the spinal fusion model for bone graft development, bone grafts are widely used in orthopedic and reconstructive surgery. Our hope is that what we learn in developing these grafts will not only relieve America's back pain, but have wide applicability for bone regrowth after trauma, cancer, or other degenerative diseases. Thank you.
Trevor, thank you. That was fantastic. Are there any clarifying questions from the judges? If not, and please speak up if I've spoken too soon, I would like to introduce our next speaker, Sarah Grace Dennis-Little. Sarah Grace received her Bachelor of Science in Biomedical Engineering from Tulane University and her PhD in Bioengineering from Clemson University. She is currently a postdoctoral scholar at the Medical University of South Carolina and is also pursuing an MBA. Today, she will be telling us about her research improving radiotherapy outcomes for cancer patients. Sarah Grace, whenever you're ready, you can begin.
Thank you so much for having me today. You have cancer. 3 words none of us want to hear, but unfortunately, most of us know someone who has. Over half of cancer patients receive radiotherapy treatment. While extremely effective in killing the cancerous tumor, radiotherapy has a cost. It also damages nearby healthy tissues. Radiotherapy protocols depend on a delicate balance between effectively diminishing the tumor while also limiting the toxicity to normal healthy tissue. Radiation toxicity affects 50% of treated cancer patients with dangerous side effects such as difficulty swallowing and breathing. Radiotherapy is so effective because it uses a high energy source to kill tumor cells by directly damaging their DNA. In this process, the high energy radiation beam also produces a burst of free radicals, reactive molecules which continue to attack nearby cells. These free radicals, however, do not discriminate between healthy cells and cancer cells. They attack and kill everything in their path. In fact, free radicals account for 2/3 of all radiation damage. However, when these free radicals attack healthy tissues, it results in radiation toxicities. This secondary damage is just accepted as the cost of radiotherapy. There are no current treatments to prevent the damage to healthy tissues surrounding the cancerous tumor, but it doesn't have to be this way. Our lab investigates how we can best protect healthy tissue during radiotherapy. Our goal, hit the tumor with a radiation without sacrificing the surrounding healthy tissue. We found out this is possible. Our lab developed a molecule, hiraloximer, that physically binds to the cell membranes of healthy tissue and shields them from the damage of hostile free radicals. Hiraloximer in no way interferes with the ability of the high energy radiation beam to destroy the targeted tumor. We investigated hiraloximer in small animals, specifically rats. We exposed the animal's right lungs to a single dose of a radiation known to cause lung injury. With no other treatment, the rats became extremely ill and developed lung damage, not just in the targeted right lung, but in the healthy left lung as well, seen in the bottom left image. In a human, this type of damage causes difficulty breathing, and if untreated, can become so severe supplemental oxygenation is required, or even death of the patient may occur. When we gave the animals a dose of hiraloximer prior to irradiation, we found a significant decrease in damage to healthy tissues surrounding the tumor. See the bottom right image where the lung injury is limited to the tumor area. The reality is that cancer will continue to be one of the most destructive diseases facing modern mankind. We don't have a cure yet, but we do have a way to improve treatment and save more lives. Thank you.
Thank you very much, Sarah Grace. What a great talk. Do our judges have a clarifying question?
Sarah Grace, Joel again. What is the mechanism for the identification of healthy cells?
So it actually binds to all cells via a hydrophobic homing mechanism to the cell membrane. It's just that the high energy radiation beam can still penetrate and damage the DNA of the cancer cells and is equally effective
Thank you.
in treating the tumor and reducing it.
Thank you.
All right, thank you very much. And just a reminder to our audience, if you do have questions, you can either hold those to the end or you can type them in the Q&A box as we go, and we will get to them in the Q&A session. We will next hear from Lishu He. Lishu received her Bachelor of Science in Biology and Visual Culture from Syracuse University. She is now pursuing both a PhD in Pharmacology and Toxicology at the Medical College of Wisconsin and a Master of Science in Law at Northwestern University Pritzker School of Law. Today, she will share with us her dissertation research on DNA damage repair in pancreatic cancer and epigenomic-based therapeutics. Lishu?
Thank you, Jess. If you consider our DNA sequences as a text or an instruction manual that explains how to make a human body, epigenetics is as if someone's adding punctuations or marking up the text in different ways. There are different types of punctuations and marks you can add or remove, and each helps us understand information in a certain way. For example, and this is a real life story, by the way, the simple addition of a comma in my text or family group chat can make the difference between a great meal together and an awkward conversation at a dining table. This is also the theoretical underpinning of my studies. I focus on, in the context of pancreatic cancer, the addition of modifications onto the subunits of DNA known as histones. These histone modifications can affect gene expression that might be important to development of pancreatic cancer. And gene expression essentially is just the process of going from DNA to a functional protein product. Again, lending from our analogy just now, I studied proteins that are members of a molecular editorial team adding common text to a DNA text. They're not changing the exact letter by letter content of what was already written, but they're modifying the way other downstream markers integral to our body functions might interpret that text, or if they're skipping a section of the text altogether, all of which eventually translates to changes in gene expression. As for pancreatic cancer, to this day, despite all the research that has come before us, it is still incredibly lethal with a barely 10% 5-year survival rate. And yet, few groundbreaking discoveries in therapeutics for improved patient outcome have occurred in the last 30 years compared to other cancers. So our challenge and my mission is to develop novel therapies. I am unwrapping how addition of histone modifications may contribute to the initiation and progression of pancreatic cancer, and how we can harness that mechanistic knowledge to develop improved therapeutic strategies. So far, we actually found that inhibiting some molecular workers, facilitating addition of histone modifications, actually suppressed pancreatic cancer cell growth by disrupting the cancer cell's ability to repair DNA damage from various sources, which normal cells usually could take care of. Now, epigenetics, specifically histone epigenetics, has only boomed in popularity in recent years. So this is guaranteed to keep generating headlines and catching the public's interest. And this very ability of epigenetics to fill gaps in our understanding of human health and disease gives me great hope that my studies delving into the epigenetics of histone can help us identify treatment strategies previously unknown for difficult to treat diseases such as pancreatic cancer. Thank you.
Lishu, thank you for another highly informative talk. Do our judges have any questions? If not, I would like to introduce our next speaker. Next up, we have Chris Ma. Chris is a postdoctoral fellow in the Department of Family Medicine at the University of Washington. She is trained as a clinical psychologist and earned her PhD in clinical community psychology from DePaul University in Chicago. The focus of her talk is on training primary care psychologists to improve access to and quality of mental health services. Chris, whenever you're ready, you can go ahead.
Thank you. Different from many clinical psychologists, I work in primary care clinics where I see patients that only have access to primary care. I remember there was a 39-year-old musician referred to me by his primary care doctor for post-traumatic stress disorder. When he completed the treatment and recovered, he dedicated his new album to me and said, "I have been looking for a therapist for 11 years, and I'm so glad to have met you here. This album would not have happened without your care." Making mental health services more accessible in primary care can really save people's lives. In the US, more than half of the individuals receiving mental health treatment are only doing so through primary care, but doctors often do not have time and skills to treat these patients. Referrals to community mental health services also tend to be very difficult. Evidence has shown that integrated models of care where mental health providers working with primary care doctors can improve mental health services in primary care. However, only 1/3 of primary care practices in our country have a mental health provider working with them. One of the biggest barriers is the shortage of mental health workforce trained to work in primary care. My research tackles an important question of how to effectively educate, train, and attract future psychologists to work in primary care. Psychologists account for a substantial portion of our mental health workforce, and the number of psychology graduates continue to grow each year. While psychologists generally have expertise in assessment and therapy, working in primary care settings require a unique set of skills that need to be taught beyond the traditional psychology curriculum. My research identified the following best practices from high-performing training programs that have successfully prepared psychologists to work in primary care. First, training programs orient psychology trainees with extensive onboarding to the culture, context, and function of primary care. Trainees learn the different roles and responsibilities as part of primary care teams. Second, supervisors in these training programs model frequently for trainees on how to collaborate with doctors and provide structured environment for trainees to practice their skills. Third, supervisors offer a psychologically safe space for trainees to have open dialogues to ask questions, share their struggles, and develop a positive professional identity in primary care. So what do we do now with this information? With funding and resources, these best practices can be implemented in more training programs to improve better access and quality of mental health services for a broader population. Growing a primary care psychology workforce is to change our fragmented healthcare system into one that truly serves the purpose of population health and health equity. Thank you.
That was fantastic. Thank you so much, Chris. Are there any questions from the judges? If not, our next speaker is Andrea Morgan. Andrea is from Southern California, where she began her research in the field of addiction. From there, she became interested in the science of pain and trauma. She is currently a fourth-year PhD student in behavioral and systems neuroscience at Oregon Health and Science University, where she is investigating non-somatic influencers like light and scent and the effects they have on chronic pain disorders. Today, Andrea will discuss how light contributes to chronic migraine pain. Andrea, whenever you're ready.
So a lot of people don't like pain, but we feel it anyway. So what's the utility of it? Well, acutely, pain alerts us to tissue damage as well as the need for medication and rest. Ultimately, it encourages us to protect ourselves. Chronic pain, disorders like migraine, is different though. It doesn't provide information that's useful, and it isn't necessary for survival. It's just pain for the sake of pain. And this is why it's important to understand how chronic pain develops and progresses, but that's not the easiest task. One reason chronic pain can be difficult to study and treat is because it's not uncommon for people with chronic pain to develop something we call sensory hypersensitivity. What that means is that sensory signals, things like light, for example, somehow become amplified to the point where they're actually able to exacerbate already existing pain. But it's not like these people are staring at the sun and then experiencing pain. This is everyday levels of light, like you'd find at home, in an office, or a store. Nothing we have reason to believe would exacerbate pain. For the longest time, doctors and researchers knew this was happening in people with migraines, for example, but it's never been clear why, even though around 80% of people with migraines experience this. This misunderstood symptom has had doctors thinking that this pain was the result of everything from anxiety to psychosis, but that's starting to change. A couple of years ago, my lab discovered that pain facilitating and pain inhibiting neurons in this part of the brainstem, in the middle panel with a purple circle around it, act the same in response to light as they do in response to other things that cause pain. So in this area of the brain, the response we have to cutting our finger is the same one that people with migraines might have in response to light. This was huge. What this meant for all those people dealing with this sensitivity to light is that their pain is as real as any other pain. But this data came with a ton of questions. I mean, we have this evidence that light can really exacerbate pain, but how? And that's where my work is at now, trying to understand how light, or how the brain allows light to get from the eye all the way back to the brainstem to exacerbate pain. I do this by working with animals that have migraine-like headaches, analyzing pain-related behavior, and by doing immunohistochemistry, which is just a fancy way of saying that I look at where in the brain there's evidence of activity that's specific to the animals with the migraine-like headaches. We now have evidence that brain structures that are involved in other types of pain, like the rostral-ventral medial medulla, are also involved in this response to light. Knowing this information will allow us to describe which structures and connections in the brain make possible this heightened sensitivity. And we call these descriptions of structures and connections in the brain neurological circuits. Once we can describe the neurological circuit, we'd have a much better idea of what to target in designing treatments for this kind of pain. Some have said that this type of pain is all in the patient's head. Based on our data, I think my lab would agree, but we'd add that it's also a bit in the brainstem as well. But to be fair, that's kind of true of all pain.
Thank you, Andrea. I know we all learned something from there, and I would like to point out that this beautiful drawing is done by Andrea herself. Joel, were there any clarifying questions from the judges?
Yeah, Andrea, thank you. Does the same region of the brain impacted by other types of stimuli like sound or touch or any other sort of stimuli?
So that's actually another direction my work is taking is into the olfactory system. And so we know light so far. We're trying to get into olfaction right now. So I hope to have an answer for that question very soon.
Thank you.
And this is cough. What model organism is your lab working in?
Frog dolly rats, males and females.
Thank you.
All right, thank you, judges. We will next hear from James Quinn. James received his Bachelor of Science in Biology from the University of Sussex and his PhD in Medicine at the University of Manchester. Now he is a Neurology Research Fellow in the Alzheimer's Clinical and Translational Research Unit at Massachusetts General Hospital and Harvard Medical School. Today, he will discuss his research on neuropeptides in Alzheimer's disease and how these proteins may help inform future diagnostics and treatments. James, you may begin.
Imagine looking up at all the stars in our galaxy. Really, you are looking back in time, as light takes 2.5 million years to reach you. Now, our brains contain approximately 100 billion nerve cells, equivalent to the number of stars in our galaxy. And when you count the number of connections between our nerve cells, also known as synapses, multiply that number by 40,000. Big, right? My name is James Quinn, and I crossed the Atlantic to research Alzheimer's disease, the leading cause of dementia worldwide. Much like stars, the nerve cells in our brain hold a window into the past. However, in dementia, our synapses start to break down, resulting in the loss of nerve cells, leading to a gradual decline in memory, issues with daily activities, and loss of independence. Someone who has died from Alzheimer's disease has lost roughly 10% of their brain, equivalent to the weight of an orange. Alzheimer's disease can only be accurately diagnosed after death. So I want to understand why this happens and develop new ways of diagnosing this happening in real time in people living with Alzheimer's disease. Improving Alzheimer's diagnosis will increase our chances of identifying new therapeutics like aducanumab to treat Alzheimer's disease. I study neuropeptides, an important signaling molecule in the brain that travels between nerve cells through synapses. We and others have discovered that some neuropeptides are decreased in the brain and cerebrospinal fluid from patients who had Alzheimer's disease. We collect the cerebrospinal fluid, which comes into direct contact with the brain by performing a lumbar puncture, also known as a spinal tap. These neuropeptides are really exciting, as they may allow us to study synaptic health in real time in healthy patients and in those living with Alzheimer's disease. However, they are not very abundant at all and really represent finding a needle in the haystack. I develop ultra-sensitive techniques to detect these neuropeptides in the cerebrospinal fluid. To do this, we use a technique called mass spectrometry. Here, we add the cerebrospinal fluid, heat it up to turn it into a gas, add some ionizing charge using an electron gun, accelerate the molecules through an electric field, and based on their size, they will separate out, which allows us to detect the individual ions. Herein lies the brilliance of mass spectrometry. It breaks apart the haystack to help find the needles. Using mass spectrometry, I've identified that specific neuropeptides may play a role in the progression of Alzheimer's disease. By identifying and tracking neuropeptides important to Alzheimer's disease progression, we are unlocking promising new avenues for diagnostics and therapeutics. My work has the potential to become an important and accurate diagnostic test for the health of synapses in Alzheimer's disease and other diseases that affect the brain. We also hope to develop new treatments targeting these neuropeptides to help protect synapses, protect neurons, and keep your brain healthy, with the aim to keep those stars shining to help you make the most of your golden years. Thanks for listening.
James, thank you for that wonderful talk. Any clarifying questions from our judges?
James, are these human samples that you're using? Or
the thing we're doing is human brain tissue that we get from a brain bank on our building, and then we collect the CSF directly. We're running a few different clinical trials, so we get CSF from those. So we have a few different neurology clinics that go on at MassGen, and we can take from there.
Great, thank you.
Excellent, thank you. Our next speaker is Faiza Siddiqui. Faiza completed her bachelor's and master's degrees in biotechnology in Delhi, India. She then completed 2 postdoctoral fellowships, one at Pennsylvania State University and the other at University of South Florida. Currently, she is a research associate in the Department of Internal Medicine at USF. Faiza will share with us today her research on anti-malarial drug resistance. Faiza, you may begin.
The largest, sorry, you can hear it. The largest killer of children is malaria that kills a child every 30 seconds. Each day, 3,000 children die because of malaria. In total, around 300 to 600 million people are infected with malaria each year, leading to over 1 million deaths in Africa alone. WHO reports that these families in Africa, they spend a quarter of their annual income for malaria treatment. So it does not only has its toll on the lives lost, but also on the economic, social, and intellectual output of these families. Now, in absence of a successful vaccine, we have to rely on anti-malarial chemotherapy or anti-malarial drugs. WHO currently recommends artemisinin as the frontline treatment to treat malaria in combination with other partner drugs. We can understand or appreciate the importance of this drug by the fact that Professor Yu Tu was awarded the Nobel Prize in Medicine for her discovery of artemisinin years ago. So artemisinin was treated as the hero for malaria treatment for a long time until 2008, where parasites started surfacing, showing resistance to this drug in Southeast Asia. What makes these parasites resistant is the question that we are trying to address. We found some novel mutations in a propeller protein that show association with artemisinin resistance. It does so when we introduce these mutations in sensitive strains, they become resistant by providing a better response towards the stress that is induced by the drug. In addition, we also found mutations in hemoglobin digestion enzyme that is associated with artemisinin resistance. That makes sense because hemoglobin digestion is essential for artemisinin activity against malaria. So artemisinin resistance actually involves multiple partners in crime. And identifying, validating, and characterizing these mutations is extremely essential to monitor as well as contain the spread of drug-resistant malaria from Southeast Asia to Africa that can be absolutely catastrophic. This will also help us in designing more intelligent and better informed drug policies. Thank you for this opportunity.
Thank you so much, Faiza. That was a very informative presentation. Do our judges have any clarifying questions? Okay, excellent. Our final speaker for the Flash Talks competition is Afra Toma. Toma received her Bachelor of Science from Florida International University and her Master of Science in Biomedical Engineering from the University of Florida. She is now a PhD student in the Joint Biomedical Engineering program at Georgia Institute of Technology and Emory University. During her talk, she will discuss her research on biomaterials to promote oral wound healing in pediatric patients. Toma, whenever you're ready.
Oral facial clefts occur during the embryologic development when the tissue of the lip or palate do not form properly. It is considered the most prevalent congenital defect occurring in 1 to 1,000 live births. And current gold standard treatments include using human donor tissue to restore the structure and the functionality of the oral cavity. However, such surgeries can also carry the risk of infection and allograft rejection, causing adverse healing. And up to 60% of these procedures fail, leading to oral nasal fistula shown on the left, which is an abnormal gap in the roof of the mouth leading to the nasal cavity. The ONF affects the child's ability to eat, talk, and thus the overall quality of life. And such high ONF reoccurrences require surgical revisions, speech therapy, and orthodontic maintenance, which can total up to $697 million in lifetime cost in the US in an annual wound care market of $22 billion. Therefore, our lab seeks to promote ONF healing using an immunomodulatory approach by re-engineering a clinically available drug called FTY-720, which is FDA approved to prevent an autoimmune response in patients with multiple sclerosis. By fabricating a biocompatible scaffold that's coupled with FTY-720 shown in the center, we can harness the innate wound healing system, examine the role of immunomodulation in ONF healing, and characterize the delivery of FTY-720 to restore intraoral homeostasis. Therefore, we hypothesized that by delivering this FTY 720 loaded polymer scaffold following a cleft palate repair, we can enhance the oral wound healing through immunoregenerative contributions associated with tissue modeling and maturation. Therefore, our lab, using both qualitative and quantitative biological techniques, are evaluating healing outcomes using microscopy images, histological staining, and immune cell profiling. Our results have shown enhanced healing following treatment with FTY 720 with tissue closure in our Miron models by day 7 post-injury when compared to control, and an increased recruitment of pro-regenerative immune cells through histological and flow cytometric analysis. We continue to demonstrate effective oral wound healing using FTY 720 as an immunotherapy, and we will further compare outcomes to that of human benefits, evaluate efficacy in large animal models, and proceed to the commercialization to provide greatest clinical relevance. This will allow us to prevent ONF reoccurrences in children following surgical intervention and also provide them an efficacious treatment option for orofacial clefts.
Chelma, thank you so much. That was wonderful. And do we have any questions from our judges? If not, I would just like to thank all of our competitors once again. That was just an absolutely terrific presentation and just beautiful science communication. I'd just like to extend a huge thanks again to everybody who has given a flash talk today. I think for us in the audience, it was very instructive of the kind of talk that we would want to give to somebody who may not know everything about our research. So I think these were all just accessible, very well explained, and I know that I learned a lot. Obviously, I cannot be an expert in every single one of these topics. In fact, I'm not an expert in any of these. So for me to be able to learn so much from each of your talks is a real testament to how excellent each one of these talks were. And thank you all in the audience for listening, paying attention, submitting your questions. So now we are going to take a short programming break. So if you would like to visit the networking lounge to connect with each other, chat, video chat, you can connect with us on Twitter using the hashtag RA21Summit. And then don't forget to log into your workshops at 3:25 Eastern Time and enjoy the rest of your day.